Okay. So thanks, Mark Seidler, for the introduction, and welcome to this webinar from BioEcho Life Sciences, where I would like to go through our product portfolio, our technologies, and give you some application examples for each of the products and the technologies that we currently have. So I apologize. I recently caught a slight cold, but I hope you can still understand me good enough in order to follow here. So single-step isolation of inhibitor-free DNA for unmatched sensitivity in PCR detection is the topic, and what I would like to show you in the next approximately 30 minutes is a brief introduction to BioEcho. Who are we? Where do we come from? And then go to the technology part. I would like to introduce to you our EchoLUTION technology, which we call the next generation of genomic sample preparation. And why we call that next generation, I will try to show you. It's a different approach to nucleic acid isolation. And I will compare our technology, especially to the traditional bind, wash, elute methods that you are probably familiar with. As I said, I will show application data for genomic isolation from blood, from cells, and from tissue, and also from the solutions for our RNA and DNA workflows that we currently provide. And this comprises sample stabilization, DNA cleanup, concentration of nucleic acids, and analysis of nucleic acids. And in the last section, I would like to show you how BioEcho products bring sustainability in molecular biology labs. So to start with, what we'd like to do is develop novel and best-in-class methods and processes for nucleic acid isolation, concentration, stabilization, automation, and processing. And whenever we launch a product, it should be fast, it should be simpler than what is available so far, and it should also be sustainable. So this is a snapshot on the team. So on the picture, you see the founders, Marcus Miller and myself on the right. Marcus and I have both been with QIAGEN for almost 20 years, each of us, where we develop many of the products that currently are marketed in the genomic sample preparation area. And our team is then further comprising of marketing and sales experiences. We do a lot in online marketing, web design, and of course we have a research and development team that is producing and developing all the products that I'm going to show you. So we have been founded in 2016. We currently have 9 employees, which cover the research, the development, the production, as well as sales and marketing areas. We have been funded by the team of the founders, by private investors, and by a crowd investing campaign. We are located in Cologne, in the Bio Campus, in the north of Cologne, with a nice view to the dome. And we are currently represented by distributors in the countries that are listed below here. And in Germany, it is SERVA electrophoresis. So I probably do not need to tell you, but I do anyway, because I have that slide, that molecular analysis in several applications are strongly increasing. And there is the research, genomic research, forensics, food testing, veterinary, pharma applications, and of course, most important or most popular molecular diagnostics, where RNA and DNA are used to do certain analysis in the personalized medicine area, in the cancer diagnostics, and in the infectious disease areas. And for basically all of these applications, it is a requirement to, as a first step, isolate RNA and DNA from various samples, which includes plasma, blood and serum. It includes tissues of various sources, including forensic samples, stabilized tissue as well, FFPE embedded, cell cultures, it includes plants, includes bacteria, microorganisms, food samples, forensic samples as well, and also paleontology samples. So this is now a comparison of the workflow that you are probably familiar with, and this is what we call silica or bind, wash, elute workflow, which is, as it is marketed by companies such as QIAGEN and all the others. And I don't want to go into all the steps here. I just want to show you that this workflow here is relatively complex. So in the first step, you lyse a sample under denaturing conditions, and then you bind it to a membrane and so on. So this workflow is not only complex, it also needs toxic and denaturing reagents in high concentrations, guanidine salts, for example, and also ethanol. It does introduce by the binding, by the adsorption, desorption steps, DNA sharing, and the reagents that are used in high concentrations may cause impurities in the DNA or RNA product. And all these steps requires a lot of plastic materials, and associated disposable costs are produced. So on the other side, our workflow, the EchoLUTION workflow, is significantly simpler. Also here, of course, you start with a denaturation, with a lysis of a sample, but not under denaturation, under the denaturing conditions, but under physiological conditions. So this is a significantly simpler workflow where you, after the lysis, you add your lysate directly onto a column that has been prepared in the meantime. Do a single centrifugation step, and then you have your DNA in the flow through of the column. And all the things that you do not want, like cell debris, you see that here when a blood sample is processed, you see the hemoglobin on the column. So the column holds back all macro molecules and also low molecular weight molecules that may inhibit or affect downstream applications. So as I said, it's the workflow works under physiological conditions. So native buffers are used. The result is high molecular weight DNA with unmet purity, and the simplicity and the low number of steps leads to the fact that we produce 70% less plastic waste and no organic solvent waste in the lab. What I would like to show you in this video now here is how we have come up with a solution to load a sample on a column, and this is shown here. We use the so-called BioEcho cap puncher. This is what you see here. It has a sharp pin that can be used to punch a hole into the lid of a column. So there is no need to unscrew that spin column. You simply place it into a tube and then punch a hole into the lid. Usually you can do that much faster, as it's actually a few seconds thing that you do. This is slow motion here. So now you see the cap has been pierced, and what you then do once you have done your lysis, you simply pipette the lysate directly onto the column, and this is already ready. And then you do that 1 minute centrifugation step, as you see here, and then the process is done. So there's only 3 minutes hands-on time. It's extremely simple. You can have a second look at that video using that QR code that you see here, and which is also displayed on all our protocols. So now, since we started with the blood DNA kit, I would like now to go and show you what the results look like if you purify genomic DNA from a from the EchoLUTION blood DNA kit. So the sample input is currently up to 60 microliters. We are pretty soon going to increase the input to probably 100 microliter, which works fine as well. So you can use whole blood. That can be EDTA, citrate, or heparin blood. It can also be used to isolate genomic DNA from dried blood spots on FTA cards. Preparation time is 25 minutes, and the yield that you can expect from 60 microliter is in the range of up to 2 microgram of DNA. Now that has been done in comparison with a silica kit here from 2 donors, and when you process 60 microliters with the silica kit, the extraction efficiency of the EchoLUTION kit is significantly higher than with the silica kit. On the second gel picture, you can see that genomic DNA cannot be isolated from human, but also from animal blood with equal efficiency. The purity is shown here. You see the spectrum of silica prepped DNA, and you see the orange and the red curves that are indicative of some impurities that are contained in that prep, whereas the typical example of an EchoLUTION preparation is that you have a flat line and only the absorption at 260 nanometers is seen. Also, if you load more samples, this is then still true. So that DNA that you purify with that blood kit can be applied to basically all applications like sequencing, PCR, and in areas like cancer profiling, infectious disease, and genomic research in general. On the next slide here, what we did here, we simulated a bacterial infection in blood. So what we did, we spiked DNA from a pathogen into human blood, and then processed the blood with a silica kit and with BioEcho in parallel, and then detected it via qPCR. So what you see here is always an overlay of the BioEcho kit, and in red the amount of DNA that we had spiked, and this is kind of the control. And this overlay implies that there's a very high recovery or sensitivity of the DNA from the blood, whereas the green lines, which come from the silica prep, they always lag a little bit behind. So this is in numbers approximately 1 to 2 threshold cycles where BioEcho prep is detected earlier compared to a silica prep. So one can say that there's a more robust detection of nucleic acids made possible once you prepare your DNA with the BioEcho EchoLUTION kit, and in fold sensitivity, there's a 2 to 4-fold difference in sensitivity detection of DNA in blood. So BioEcho is more sensitive and more robust. It can also be done very reproducibly. What you see here is 36 preps that have been done in parallel, and this is basically only to show you that this can be easily done and very, very reliably being done. The CV is below 20%. I'm now coming to a second application and second kit as well. Is the EchoLUTION Tissue DNA Micro Kit. It's called micro because you can load or process as low as 0.1 milligram, and it can be used up to 10 or 20 milligram of human or animal tissue. Preparation time is 37 minutes, whereas with most other kits you need 2 hours or even an overnight processing of the samples. The yield with our prep is up to 50 microgram, and it can be done with basically every tissue that you can imagine. So there is basically no problem that we ever. That is also holds also true for fatty tissues like brain, or for fibrous tissues like muscles. And there is a pretty linear increase in yield from 0.1 1 to and 10 milligram, which is shown here for that liver prep. Also here, the purity picture looks very similar to what we saw in the blood kit. We frequently see these impurities in silica kits, which is caused by salt or residual peptides or other impurities, whereas we are able, with our specialized matrix, to really make sure that all these impurities have been removed from the prep. So application is also here basically through the entire area of possible applications, including sequencing and PCR. This is some further technical data where in that gel you see that the chain length of the BioEcho genomic DNA is longer than with silica preps, and that could have advantages, for example, in nanopore sequencing applications. This is DNA prepared from mouse tail, and also the yield of the EchoLUTION prep is approximately threefold compared to silica, so which shows that the extraction efficiency, due to the fact that we are able to work under physiological conditions and use the optimal enzymes, the optimal proteases, for example, to extract the sample, that results in that significantly increased efficiency of extraction. Another observation that we made is that OD readings do not necessarily tell you the truth. What we did here is we prepared genomic DNA from brain tissue and analyzed that on a gel, and you see that the genomic DNA band from the silica prep is significantly lower than on the EchoLUTION. Okay, that's what we saw before, but if you then compare that to the OD reading, the photometer says that you prepare 6.7 microgram with the silica kit, whereas only 3.4 with the EchoLUTION. Well, obviously, this is misleading, and the thing is that that in silica preps you always or usually have a lot of other molecules that contribute to the OD reading, and for example, RNA is in significant amounts included in these preps, so that the OD reading alone does not necessarily tell you the truth. It should always be compared or accomplished by a gel analysis, or even better, by a functional analysis. So this further underlines the increased efficiency and performance of BioEcho preps compared to what you were used to so far. Also here, a qPCR kit, qPCR experiment has been done where we compared tissue DNA versus silica. In this case, DNA prepared from mouse tail, and we compared and if you compare the red traces, BioEcho compared to the blue, the silica traces, you again see a significant difference in detection sensitivity or DNA recovery. That is another possibility, but it's what you see is 1.6 to 5 CT in difference, and this again underlines the increase of robustness of nucleic acid detection upon BioEcho preparation technology. In order to show that our process reagents, it's probably the process reagents, or partly at least the process reagents from the silica kits that contribute that reduced sensitivity, and to show that our process reagents do not introduce any inhibition, we did that experiment where we over the entire range here included 1 microliter of the DNA that we have obtained, and then spiked a mock prep where we did not have any sample or DNA, but we just did a prep, and the resulting elution fraction, which may contain or may not contain process reagents, were titrated. And even if we go up to 6 microliter, which is more than 30% of the total reaction volume, we do not see any inhibition. So this, from this and other experiments, we can say that our process reagents are not inhibitory, and there is no surprise because we do not use guanidine hydrochloride, we do not use ethanol, we do not use any high salts at all. So it's physiological conditions. There is no inhibition by nature. And I'm now coming to the cell culture DNA kit, where you can use up to 2 million cultured or primary cells to prepare your genomic DNA, and this is done within 30 minutes. And also here it's up to 50 microgram of genomic DNA. Basically, there is no upper limit because we do not have to deal here with the binding capacity, but it's the flow through. So the DNA does not need to be bound somewhere. It just flows through the column, and everything else is held back on the column. And you see that difference in yield on the gel here, where with two different cell lines, we get a significant increase in yield compared to a silica kit. And what you, in numbers, is 11 versus 37 or 25 versus 43 microgram compared to the silica kit. This kit has been used by a customer, and this is customer data, and this customer did a titration of input from 15,000 to 2 million cells and compared the yield of the DNA, and you see that it's over the entire range pretty linear. And then he did PCR on all these samples, and you see that also the PCR works reliably irrespective of the input of the DNA amount. So there's a linear DNA yield over a wide range of input, of sample input, and a robust amplification of genomic loci from this genomic DNA that has been purified here. So this was DNA extraction, what we have so far. Now I'm coming briefly to DNA cleanup, and we are currently expanding our DNA cleanup portfolio significantly. What we have so far is the EchoClean DNA cleanup kit range, which is in a single step process, able to clean up and dissolve DNA within 3 minutes. It's basically the same workflow that you see, as you see here on the right. You have a sample added onto a column, which has been prepared by a 1-minute packing centrifugation step, and then so you load the sample onto the column, do a one centrifugation step, and then you're done. And it can be used to, for example, exclude oligonucleotides of a certain size range. Everything larger than, smaller than 50 base pairs, smaller than 100, or smaller than 200 base pairs can be removed. Phenol can be reliably removed. It can also be used for PCR and enzymatic inhibitor removal, and for desalting, of course. The yield is very high, so it's over 95%. Again, due to the fact that we don't need to bind, so we lose nothing during binding. We don't have to desorb. We lose nothing also during washing steps and so on. It's a single-step protocol, as I told you. The time is 2 to 3-fold faster, and again, there is no hazardous materials and the minimized plastic waste associated with these preparations. So we, if you consider if you use tissue and are interested in isolating genomic DNA or other materials from that tissue, we have a certain product range that covers many steps of such a workflow. So we also have these ceramic blade scalpels, which are from ceramic, so not steel. They do not rust. They stay sharp a very long time, and they can be used for a very long time with extremely high performance. We have a tissue stabilizer for stabilizing the tissue for later DNA or RNA purification from these samples. and we have then the tissue DNA microkit, as I just introduced to you. And then we have also, if your workflow requires a concentration, we have a single-step concentrator technology, which I'm going to show you in a second. Briefly, just to the stabilization reagents, which we call PurifyLater. As I said, it stabilizes human or animal tissues for a very long time, and you can use that for storage in a very safe mode. It reliably protects both RNA and DNA against degradation, and it can be scaled to your need. So this picture here shows a DNA that has been stored under unstabilized conditions. So the tissue has been stored under unstabilized conditions, whereas and you see DNA degradation, whereas there is no degradation if you store tissue at room temperature in PurifyLater. And for a long time, long-term stabilization, PurifyLater results is exactly the same as if you store the tissue at -20 degrees. So there's no decrease over basically unlimited time of storage. As I said, we have a one-step concentration technology also. So this is done by a super absorbing polymer technology, SAP, which we have exclusively. You load 200 microliter of a DNA or RNA solution on such a column, and you see that here in that schematic down here. So this is column. You add your sample in number 2, and then these super absorbing polymers, they swell. They take up water. They do not take up RNA or DNA because of the surface modification. So DNA and RNA are excluded. So once this swelling has been accomplished, after 3 minutes, you break off the seal, do a single centrifugation step, and have your concentrated nucleic acid in the flow through of that column, and you can concentrate up to 20-fold. And this is shown here for RNA in the RNA concentrator. So this is the RNA before the concentration, and this is a 4-fold replicate and shows the concentrated RNA. And then also has been analyzed by qPCR. So there's a significant difference in I think three or 4 CTs earlier detection upon concentration, which shows the concentration effect. So this is pretty reliable. And then we also have a product range which is growing as well for covering your part of or your entire RNA workflow. For example, if you work with paraffin-embedded pathologic tissue sections, you can use the EchoSafe deparaffinization solution to extract the DNA or the RNA from the sample. If required, you can concentrate your sample again with a reduced concentrator that I just showed you, and then you can analyze your RNA on a gel by using the EchoSafe RNA gel loading buffer, which is another product that we have, and you simply add that buffer, and then you can analyze your RNA on a standard agarose gel. And this is, I'm going to show you that in a minute. So this is just, again, on the FFPE deparaffinization solution. It is non-toxic, and if you and non-volatile, and so there's no use to work under the hood for deparaffinization if you use this reagent. You may use that reagent. So far, we have only the reagent, a kit for FFPE deparaffinization or for the purification of the deparaffinized tissue is in preparation. But what you can do in the meantime, can use that deparaffinization solution and combine it with a silica kit, a basal silica kit, and save approximately 30%. So if you have a look on that number here, €8.5 with a is the cost for one reaction of one of the silica FFPE kits, whereas the RNA extraction kit alone is only 5.8. So if you combine such a normal extraction kit with our EchoSafe reagent, then you come to a reaction price per reaction of 6.2 euro, which saves you 30%. So that can be used here. Now this is the EchoSafe RNA gel loading buffer. As I said, it's a reagent that you add where you simply add your DNA or your RNA to, and then you can analyze your RNA on a standard agarose gel. If you would do that with a standard gel loading buffer, then you get RNA degradation, but the RNA is protected from degradation if it's run in that RNA gel loading buffer. So no more need for toxic formaldehyde formamide gels. No need to run gels under the fume hood. Just use a single standard agarose gel to analyze your RNA. And you can do that in parallel on the same gel with DNA analysis. Now, in coming to the end, just wanted to show you that sustainability in molecular biology lab is possible. You're probably aware that you produce, if you do RNA or DNA extractions, you produce a lot of plastic, you produce a lot of organic solvents, but if you use BioEcho kits, you can save a lot in this respect. So what we do is we consequently follow a philosophy of developing sustainable products. You save 70% of plastic consumption. You do not produce any organic solvents. You do not have any toxic solutions in general in the kits. So, and in addition, we follow an environmentally friendly packaging concept, which is plastic-free as far as possible as well. So this gel here, this picture here shows you the difference of a 250 reactions kit on tissue, on purification of DNA from tissue. This is the amount of plastic with a BioEcho kit, and this is the amount of plastic that you produce with the silica kit, plus you see the in red here, the organic solvents. So BioEcho produces 70% less, and we want to be and to stay eco-friendly and help saving disposable costs. And just want to go quickly to that slide here, just show you the end result. You can go through the details here as well. So this is a calculation that with a single test that is produced on a routine basis, you can save up to 200,000 euro per year with only this single test if you switch from a silica technology to BioEcho. And you can do your own math later on maybe and try to realize whether this is something worth considering for you as well. What is going to come in the next weeks or months? So we are almost ready to come up with a plant DNA kit. We are going to beta test that in a few weeks. We are working on a bacterial DNA kit for total DNA from both gram-negative and positive cells. We are in the final stage of developing a DNA and RNA FFPE kit. We are going into high throughput as well, so we will come up with these 96 well plates. We are in the process of producing swap kits for RNA, for DNA, and probably RNA as well from stool and other like buccal swaps. EchoClean product range will be expanded for removal of organic solvents, especially as a preparation for next generation sequencing, and also as a cleanup for RNA products. And the RNA extraction is one other big project that is currently ongoing. And by that, I would like to summarize, and I hope I could convince you that EchoLUTION is a single-step purification, which is faster, gives higher yields with the superior quality of DNA, and which is a very convenient procedure. We have products over entire workflows for DNA and RNA. BioEcho brings eco-friendliness and sustainability in molecular biology labs, and more is to come very soon. And by that, I want to thank you for your attention, and I'm ready to answer your questions. Thank you.